Hello, everybody. So recently we had a webinar with the fine folks over at Biodyne. This is a company that makes biological products for farming, and we had a live broadcast of it. We wanted to make sure that we had a recording of it. It is a little bit longer, a little over an hour or so. But stay tuned to the end. There are some great questions that were asked by some of the people that were viewing it, and we think you can get a lot of information from it. So thank you for watching, and hope you enjoy it. If you have any other questions about it, leave some comments, and we'll be able to get them answered for you. And as always, like and follow and subscribe to what we have going on here, so you'll be the first to know about anything that we have coming out. Thank you. Take it away,
Well,
guys.
yeah, thanks a lot, Andrew. We are Biodyne USA. I would like to introduce Garth Farley and Bodie Kitchell. We're going to walk you guys through some of our innovative technologies here. You can take it away, Bodie.
Yeah, so as Grant said, my name is Bodie Kitchell. I'm director of agronomy at Biodyne. A little bit of a quick background on me, I think, as it pertains to a lot of the data that you're going to be seeing here. About 4 years ago, I joined the company, and really my focus was to try to bring transparency to the biological marketplace. As you see through this presentation, I think you'll get to see just exactly that. You'll hear from Garth, who is our Chief Technology Officer here at Biodyne, and he'll walk through a little bit of Biodyne, the biologicals, what goes into it, and really just connecting human health, plant health, soil health, and really how it all goes back to biology. And really, what we would like to do on this is, I know for me, and I talked with Andrew about this a little bit before, is we like these to be interactive. So if there are questions, we'll be happy to answer those questions at the end. So if you have any of those, please save them. We'll try to cover everything here. But with that, I'm going to kick it over to Garth. And then I'll go through a little bit of what we've seen from a field perspective.
Yeah, thanks, Bodie. This is Garth Farley. Want to thank everyone for joining us, and appreciate Bass Hybrids to give us this opportunity to talk about our story and our technology. And I think the power of that and the benefits of this technology when it comes to growers and the profitability and the sustainability that we're all endeavoring for. So about Biodyne, as they said in the intro, we're a family-owned and operated company, been around for 3.5 decades. And, ultimately, we're a science-based discovery company, and we're focused on the power of nature, fundamentally through the use of beneficial microorganisms and building tools that are useful in different environmental applications. And we'll spend most of this talking about our agricultural tools and the ways that they can be utilized. Next couple of slides are just going to be a little bit of a history lesson on who we are, to give you some context, based on what brought us to this point. So Biodyne was started by Dr. Fred Farley back in 1989. And Dr. Fred Farley is a PhD microbiologist. He's also a master's in plant physiology. And, ultimately, he wanted to leverage those skill sets to have a positive impact on the environment. And as we got started, we quickly understood that nature has created a lot of tools that could be very useful for specific applications. And those are naturally occurring beneficial microorganisms. So back in '89, he got the idea of taking naturally occurring beneficials with specific skills and creating teams to have impacts in the environment. And where we started was in bioremediation. So what that is areas where there's a contamination. Let's say there's a diesel spill. or there's a pipeline that's been leaking oil and hydrocarbons into the soil, he introduced this particular technology, this team of naturally beneficial microorganisms that could remediate, could use that hydrocarbon, use that diesel as a food source to naturally clean up the contaminated soils or waters. And that was kind of the beginning of our journey as we began to discover and explore the power of those specific naturally occurring beneficial microbes. And then over the years, we began to build on that technology through isolation and discovery of specific individual microbes, and then understanding them not as individuals, but what they can do together as a team. So began that journey in hydrocarbon remediation, and then we moved into other complex organics. So think chloride compounds, think herbicides, pesticides. And again, fundamentally, it's the same strategy. Use what nature's already created, what she's given us, but use it as a technology and a tool to solve some environmental challenges. And then around 2000 to 2011, we began to start thinking about agriculture and how those technologies could help find profitable solutions to cash croppers. And back in 2012 was when we really began to understand the capacity of our tool set, our toolbox, and really began to commercialize it at scale. I think back in 2012, we were fortunate enough to get on about 1,000 acres and really see what our products can do in the real world. And over the decades since then, you know, our products are currently on millions and millions of acres in North America and ultimately around the world through our partners in different parts of the world. So it's basically a science story. We're a science company, and fundamentally, we believe in data-driven results and the proofs in the pudding. And that's what we've endeavored to do over the last 3.5 decades. So something that's really unique for us in this space, and as Andrew kind of had alluded to with Bass Hybrid's search for this type of approach, these type of tools, it's a bit of a Wild West. And for us, we've got a little bit of a different approach, meaning we're vertically integrated. We don't purchase our microorganisms from anyone else. They are microbes that we discovered using our discovery capabilities. We don't ask anybody else to produce the final products. We do all of that in-house. And something that's really fundamental about that, I think, is it gives us a level of control over our process that gives a real focus on quality. You know, and quality for us is really about consistency. So we really take care to produce our products in a way that ensures that each gallon that's produced is going to deliver the capabilities that we've seen in the field, in the lab, but do it consistently because of that process. And this is something that's really unique about us. If you're familiar with the microbial space, there's a lot of products out there making a lot of claims, but ultimately we are in control of our own destiny through the discovery, through the creation of the products, and then the delivery of those products. And again, I want to emphasize the fact that that gives us a level of control over the quality, which is shown in our ability to deliver it year after year and the benefits that we've shown to our customers. And again, that's about the team approach, much like our products. It takes a group of folks, it takes a group of tools to deliver these science-based solutions at the scale that we deliver them with the consistency that we deliver them. And again, it's about that quality. Being 100% vertically integrated, we can manage the process from start to finish to know that it's going to meet our standards, as well as find ways to be a little bit more efficient in that process and ensure that we've got a product that will be consistent for our customers and will deliver on the capabilities that we've seen at scale across millions of acres at this point. So let's just talk a little bit about what a microbe is, you know, a little bit of a science lesson. You know, microbes are everywhere. If you were to take the weight of all the microbes in the world, they would be the largest life form on the planet. But the interesting thing about microorganisms is you don't see them. You see their effects every day. You feel their effects in your body, but ultimately you can't see them. And that's what's really powerful about them. They're kind of the unsung heroes that makes life viable on the planet. And for us, we really value that aspect of them as being fundamental in agriculture, specifically in driving the processes that create a plant, that create that plant into a fruit, and then to create that fruit into yield. And for us, it's about leveraging the power of microbes, leveraging the power of the science around those microbes, and creating tools and technologies that growers can implement without changing their production strategies, but introduce these technologies into providing a much more prosperous promotion of yield within their systems. So if you think about it, we've all heard about probiotics, all right? Probiotics are introducing microorganisms into your digestive system. Why would you do that? The fact of the matter is your digestive system is driven by microorganisms. The microorganisms are the ones that will take, let's say, that apple that you ate for breakfast and break it down into fundamental elements that then your body can absorb. Your microbiome would not be there, and you would not be able to satisfy and feed yourself without the microorganisms that are present. And what they're doing is they're taking complex structures and they're breaking them down to the simpler structures, like the nitrogens, the phosphorus, the potassiums that your body needs to create and build tissue. So fundamentally, microorganisms are driving you as a life form and creating the nutrition, the energy that you require to grow big and strong. Again, just kind of to reiterate, if you look at our bodies, they contain trillions of microorganisms. You don't see them, but you see the effects of them. And if you were to compare the amount of microorganisms in your body to the amount of cells in your body, it's about a 10-to-1 ratio. And this just gives you an idea of the absolute magnitude and the role that microbes play just within our digestive tract, just within our ability to create life and create prosper as a life form. Another interesting thing about the microbiome within our guts is, they've done a lot of research, and serotonin is a well understood biochemical material that is very important in brain activity as well as the physiological response of a human. And they found that up to 95% of the serotonin we utilize every day is produced by microorganisms in our guts. And if you look at serotonin, it's known for modulating mood, cognitive response, reward learning, memory, all positive things. Now, I asked myself when I first learned this, why would a microorganism spend the energy to produce biochemical compounds that will help me as a living being feel better? And it's a simple relationship. The microorganism wants me to feel better so that I will eat more food that then it can use as a food source to create more nutrients for me. So it's really a symbiotic relationship when you look at it. They're doing the heavy lifting when it comes down to breaking down the food to a material that my body can then absorb. And they're also releasing biochemistry that promotes my health as an individual. And that relationship is what drives life, not just in us, but as we'll get into it, it drives life in the soil systems and in commercial farming. Another material that we know much more about is dopamine. And again, it's something we've all heard about. And again, it's about decision-making, attention, memory, again, all things that promote your ability to thrive as a human. Again, up to 50% of that dopamine is produced by microbes in your gut. And again, they're doing it not because they love us, they're doing it because they get a benefit from promoting health and happiness within us, which means we're going to provide them with more material that they can use as a food source and ultimately driving our ability to be happy, function, and create tissue and bone. It's quite a unique experience to think about the benefits that we have with these little organisms that we don't see, but they are fundamental to our ability to live. And I would suggest the same thing is happening in our farm fields. So we're going to spend a little bit on this slide about soil microbes. And, when I look at this and I look at the relationships, I think about the previous slides. If you think about a plant, it doesn't have a stomach, it doesn't have a digestive tract, it has roots and root surface area. So what takes the fertilizers that we apply or the nutrients that are locked up in that soil and turns them into a plant available form, a form that then can be absorbed through those root systems? It's microorganisms. Similarly to what they're doing for us in our bodies, taking a bite of an apple and turning it into absorbable nitrogen, phosphorus, potassium, microorganisms are doing the exact same thing in the soil system around that root zone. They're creating plant available nutrition through their processes of breaking down complex structures into fundamental nutritional counterparts. You know, it's almost a destruction to creation. The microbes will take something that's not available to the plant. They'll use their enzymes and their methods to create byproducts that are available to the plant, driving that system, creating that nutritional feed for that plant and allowing those roots to take advantage. So for us, this is one of our favorite slides. And me personally, you know, this is a slide that I could spend days and days talking about. You know, there's a fundamental understanding that I think we all see is the plant and the soil are in partnership. And it's a really well understood concept at this point, that symbiotic relationship. As I just got done discussing, the soil is what feeds the plant. The soil, through those microorganisms, create plant available food, but we're all aware of the process of photosynthesis, water, CO2, and sunlight creating sugars, creating carbon, and fundamentally that plant will create those materials and through its root system, introduce it into the soil, thus feeding the microorganisms the fuel that they need through carbon so that they can go about their primary function, which is taking complex, unavailable nutrient sources, breaking them down into a plant available form. And from that perspective, if you think about that relationship, no one can control the weather. But as we'll get into a little bit later throughout this Zoom, growers can have a direct impact on that root zone, on the soil. And we believe that with our approach, by introducing specifically designed teams to optimize, almost like a probiotic for the soil, to optimize that symbiotic relationship, to drive that process of taking unavailable nutrients, converting them into a plant available form, you can really find an opportunity to not only find profitability in your growing system, but also inherently drive yield potential. If you have a well-functioning soil system through a well-functioning microbiome, through microorganisms specifically partnering with that root zone, you're going to get a much more efficient system. You know, you think about, you put a seed in the soil, right? What's feeding that seed? That seed germinates. What's feeding that root zone? You're feeding the root zone through that soil. So the plant is growing because of that soil. When you make an application of a fertility, you're not necessarily feeding the plant, you're putting that fertility in a position, i.e. the soil, so that then it can be made into a plant available form through the microbiome, through the microorganisms. And if you can really activate that association between the plant and the soil and the plant, you can drive such significance in efficiencies in that production. And if you recall earlier on, I talked about in our guts, the microbes that are releasing serotonin and dopamine to try to promote our well-being and our health. The microorganisms in the microbiome around that root zone are doing the exact same thing. They're not just producing nutrients, they're producing biochemical compounds that are promoting resistance to abiotic stress, promoting opportunities for that plant to be successful because, again, through photosynthesis, those microorganisms are going to get that carbon that they need to function as energy. And in return, they're going to provide plant available nutrition through their enzymatic activities. And if you think about It's the circular economy. The microbes are rewarding the plant through their activities. The plant's rewarding the microbes through their activities. And that partnership is really the key to yield potential. Nowadays, if you look at commercial agriculture, the way that that soil is treated is not as if it's alive, but as if it's just a transport mechanism. But what we've learned over the last decades is that if you optimize that soil system, if you optimize that association with naturally occurring beneficial microorganisms, you can really drive the efficiencies in that production system and really promote a plant that is going to be as healthy as it can be and really want to produce that fruit, which is what that seed wants to do. You know, the seed just wants to create more seed. That's its goal. And the soil is what really drives that. And the soil is what we as growers can have a significant impact on through the introduction of materials and the choices that we make. So we just was talking about this is the carbon cycle. And, you know, it all starts with the photosynthesis, right? CO2, water, sunlight, captured by those solar panels, which we call leaves. Then that's converted into carbon, basic carbon. And the majority of that carbon, in some cases, is returned into the soil via the root system. And then that carbon is utilized by the microorganisms in that root system as a fuel source so that they can then do the microbial activity. And you look at it, I like to consider it a circular economy, right? The microbes get the carbon, they produce plant available nutrition, they produce biochemistry that promotes plant health, and they encourage that plant's ability to grow more solar panels, to capture more sunlight, to produce more carbon. And the great thing about our approach is if you optimize that soil system, if you get a nice diverse population of beneficial microorganisms in there, they're not just producing plant available food, biochemical products that promote plants' health, but they're also releasing CO2 into that canopy. And as I said, it's CO2, water, and sunlight that drives photosynthesis. And by creating a beneficial environment via microorganisms in that soil system, you're really capturing that carbon in that system, but you're also releasing it through CO2 respiration back up into the canopy, which will drive that cycle, create that circular economy. And that's where the efficiencies come in.
Hey, Grant, real quick here, go back on that slide just a little bit. There might be some terms on here that you guys maybe have never seen before, and just want to make sure that you kind of have an understanding of it. You'll hear us talk about it an awful lot. As Garth talked about, he's 1,000% correct. And you look at that first step from the photosynthetic process, and you look at the microbial activity, and you look at enzymes, right? You see enzymes. You heard Garth talk a little bit about enzymes, and we're going to go a little bit deeper in that as this presentation continues. But that WEOC, which is water extractable organic carbon, that is the food source that drives the entire biological system. The reason that that's so important is there's a lot of times that we get asked by growers is, you know, I farm a 5% organic matter soil, so I don't need biologicals, right? And for every one of those questions, there's a question on the other side that says, I have a 1% organic matter. That means that I'm not going to be able to keep alive the biological activity, right? And what we have found is if you're not familiar with Dr. Rick Haney, he was out of Temple, Texas. He developed what's called the Haney soil test. And one of the things that we use to validate what our microorganisms are doing is looking at that Haney soil test to be able to quantify and qualify the nutrient availability that we're going to be talking about. And one of the first steps in understanding is there's been data done by Penn State that looked at the R value of organic matter to biological activity as well as the WEOC to biological activity. What they found was there was a very, very, very poor correlation from what your organic matter levels are to biological activity to what the actual WEOC portion of that soil is. The reason that I think that is so fundamentally important is for two reasons. Number one, what I like to do is break out biologicals into really two categories. You have creators and you have consumers, and we're going to talk about them because we have both. And when I think about the creators are those that have the ability to take your cellulose, lignin, chitin, right, complex carbon that are not in the water extractive organic carbon form. So think about things that make up organic matter, right? There's so much material there that is not broke down for biological activity to be able to use, which is why we see poor correlations from organic matter to biological activity. So you have the creators that are doing that, breaking down the complex carbons into the available carbon. And then you have the consumers. And the consumers, what they do is they use that WEOC to grow their populations, to feed on as a food source, and then to produce the enzymes. Again, you heard enzymes talked about in Gar's presentation, to be able to produce the enzymes that facilitate the nutrient availability, right? Oftentimes, a lot of the fertilizer that's applied in our soils goes in as inorganic chemistry, right? The great thing about inorganic chemistry is it's immediately available for plant uptake. The bad thing about inorganic chemistry is it has the ability to complex with counter ions. So if you think about phosphorus, for example, right? The counter ion to phosphorus, so phosphorus being an anion, so a cation would be the counter ion to that. So you think about what exists in your soil as a counter ion to phosphorus, that would be calcium, magnesium, aluminum, iron, zinc, manganese, right? Just to name a few. That means that those nutrients have the ability to form complexes to phosphorus. So you have calcium ditriphosphate. That's an insoluble bond that is not plant available. The plant doesn't take up those bonds. It takes up calcium and phosphorus separately. The reason that that's so important is, as we move into the new slide and we talk about this slide here, is carbon. The more carbon that we can put in the soil, the greater ability to create season-long, think of it as mineralization, or season-long release gets exponentially better, because what we're doing is we're tying it to a carbon molecule that biology down the road is going to consume and to release that nutrient, rather than letting our soils continue to fix those nutrients, right? So as Garth said, you know, the industry has focused on looking at the soil as kind of a dead medium, if you will, not really admitting that it's alive. And by that, we have, as an industry, we have put just pounds and tons of inorganic fertilizers. And we've actually seen that the higher we drive some of these soil test levels with inorganic chemistry, we actually have seen production go backwards, right? So as we go into this slide, this next slide, talking about the phosphorus cycle, I think it really starts to explain an awful lot of really how this happens. So today, this is university data. This is not our data. In the top right, for every $100 we spend, or every dollar we spend, however you want to look at it, we're getting anywhere from 10 cents to 30 cents on every dollar we spend on our phosphorus, right? And when you think about what you've heard so far in this presentation, I want to make sure that there's two things that are clear. Number one, we're not anti-fertilizer. We're just pro-farmer efficiency. That's number one. Number two, we obviously, as farmers, and I grew up on a farm, I'm still active in our farm today, we know that we're getting 10 to 30 cents on our dollar. We're not going after that. What we're going after is the other 90 to 70 to $90, right? How do we drive efficiency in our soils and get more on every dollar that we spend? So I think this slide does a really good job of kind of depicting that, if you will. The industry, if you have ever taken a soil test, and it's either been measured as a Bray, use a Bray acid and Olsen acid for phosphorus, or a Malic 3 extract. What you're looking at is just that center pool. So if you see what says active P, that is just a measurement of that. And for years, universities, industry, etc., that is how we've made every fertilizer recommendation that we have made on our farm fields, is just looking at that pool, without recognition that there is the pool to the right and the pool to the left. The pool to the right would be designated as plant available, right? So the number that you would use or how you would get that is that's using root exudates and deionized water as the extraction process. So that's a part of the Haney soil test, again, if you've heard it. On our soil test, we use an indicator. It's called an indicator complete. The lab is in South Dakota. Next Level Ag Labs is what it would be. So how you would see that designated would be H3A, right? So that's plant available phosphorus. On the left-hand side that's your total or fixed pool, right? So that's phosphorus that would be bound to carbon. And for example, I was looking at some soils in central Illinois last week. The soil test had 1,260 pounds in a 0 to 6 inch soil sample. 1,260 pounds of phosphorus in a 0 to 6 inch soil test, right? Total phosphorus. The Bray, which is the active pool, was only 29.9, right? And the plant available pool was 25.5 part per million, right? So you start to see how this happens. And the question that I often like to answer for folks is biology really it's not magic. It's very simple. Okay, this is what they do. Number one, the first question that biology would ask itself is, do I like my environment, right? Is the pH good? Do I have good soil structure, right? Those are two things from a biological perspective. If those two things are good, you're going to be able to facilitate pretty good biological activity. The second thing is do I have a food source? So go back to, if you think back to the one slide previous that we talked about the carbon cycle, we talked about WEOC, right? So if those two things are good, what they're going to do is they're going to grow their numbers. As they grow their numbers, they're going to continue to eat. As they consume, they then excrete those enzymes, right? And those enzymes are what facilitate the availability of the nutrients, right? So for example, if I want to try to unlock some of this phosphorus as a grower, right? If I look at this data and I think, man, $10 to $30 out of 100 or 10 cents to 30 cents on every dollar I spend isn't good enough. I want to be able to figure out how to do better with that, right? The thing that you need to be looking at from a biological company and a biological perspective is you better have phosphorus solubilization or phosphorus solubilizing bacteria, right? Putting a diazotroph or an ammonifier in and expecting them to make phosphorus available might be setting yourself up for failure or unmet expectations, right? So really understanding that the nutrients in our soil is that, as Garth talked about, he talked about the economy of circle, right? The circle economy is you got to see this, right? So we go from the total or the fixed phosphorus pool to the active pool to the plant available pool, and understand that is facilitated by biological activity and enzymatic processes. So how do we achieve this, right? Number one, keeping the WEOX high, as we've talked an awful lot about, which is the food source. Plant stimulation, you heard Garth talk about it as well. Soil feeds the plant, plant feeds the soil. Make sure that we're keeping plants healthy. The healthier a plant, the more exudates we're going to produce. Those exudates are sugars that our microbes are going to feed, right? We talked a little bit about the pH, promote mineralization, and keep a profile full of enzymes. But again, at its core, what's driving the cycle is microbes and their biological activity. I think, if I had to brag from a company perspective, I truly think this is one thing that sets us apart from every other biological company in the marketplace. What you heard Garth talk about at the very beginning was the background, right? Over 3.5 decades, right? Since 1989, we've been in the biological space. The focus early on was simply bioremediation, right? So what you need to know is what you see there in blue is the bioremediation aspect that these microbes are facilitating, right? What it is they have the ability to break down these petroleum hydrocarbons and use them as a sole energy source, right? As a grower, what you really care about is what's on the right-hand side, right? And what is on the right-hand side is simply the capabilities and how many strains are providing that specific enzymatic process, right? So we talked a little bit about phosphorus. You can see it's the fourth or fifth one down on the right. We have 6 strains that have the ability to solubilize insoluble forms of phosphorus. So again, it could be iron phosphate, that could be calcium phosphate, that could be magnesium phosphate, zinc phosphate, right? That would be insoluble forms of phosphorus that we have the ability to break down, thus resulting in driving a higher net efficiency and grabbing more dollars, right? Driving more dollars of every dollar we spend. So driving that nutrient efficiency. And that's really what biology does at its core, right? At its core, they don't care whether our plants yield 10 or 15 or 20 bushel more. They really don't. That's biology at its core. What they care about is, do I like my environment? Is there food? If the answer is yes to those 2 questions, then I'm going to produce these enzymes that facilitate these reactions. So the top one you can see, that's proteinase, right? So that is, they have the ability to break proteins down into the amino acid components, right? That is important on a lot of different levels, right? You can see the IAA there, that's indole acetic acid, that's a rooting hormone. You can see that we have 7 N2 fixers, which are diazotrophs, have the ability to fix atmospheric nitrogen into ammonia. More so than that we talk an awful lot about is the ammonification, right? Or the ammonifying microbes. What they have the ability to do is break down organic in to inorganic in through an ammonification process. The reason that this is important is I know there's a lot of buzz in the industry right now about N2 fixers, right? I think there's some things that maybe don't get shared that are extremely important. Number one is this. N2 fixers at their core, right? At their core, if they're not genetically altered, if they're not genetically modified, they will not fix atmospheric nitrogen in the presence of high available nitrogen in the soil, right? So if there's a tremendous amount, and a tremendous amount could be 60 pounds, 50 pounds, 70 pounds of nitrogen in the soil. N2 fixers aren't going to fix it simply because the demand, the energy demand that goes on that bacterial cell, they're not going to do it, right? It takes more grams of carbon per gram of nitrogen output for nitrogen fixation or biological nitrogen fixation to happen versus the plant taking up the nitrogen itself. So the reason that we talk more about the ammonification is because we know, go back to our chemistry lesson, right? Is the difference between inorganic and organic chemistry is what? It's simply a carbon molecule. That's it. That is the difference. Nitrogen that is plant available would be inorganic chemistry, right? Nitrogen that is not plant available would be organic nitrogen, right? So to be able to facilitate that breakdown, you have to have ammonifying microbes, and we have 15 ammonifying microbes. On top of that, we have the ability to oxidize unavailable sulfur, so elemental sulfur, whether it's a Tiger 90 type product, whether it's ATS that you're using. It has to go through an oxidation period or process before it would be plant available. You can see that we have a strain that is focused on phosphorus solubilization. You heard Garth talk a little bit about whether it's biotic or abiotic stress. ACC deaminase is a precursor to ethylene. Ethylene is a gaseous hormone that if the plant is under attack from a biotic or an abiotic stress, ethylene will accumulate inside of the plant. What happens as a result of that is that the plant stops sharing the sugar or the exudates. And when that happens, it slows down your biological activity. As a result of that, you're not making those nutrients available. You don't have that cycle, if you will, happening because the plants aren't getting the same amount of exudates that we were. We also have the ability, we have KSBs or potassium solubilizers, as well as zinc solubilizers, right? And I think there's a couple of things on this slide in particular to point out. Number one, is the transparency, right? You heard me talk about it. There's a reason that I came here. I will say this from a lab level, from a bench level. I will tell you that our team of microbiologists, I'm not going to say they know everything about our product because every day they're trying to learn more. but I think that they know a tremendous amount about our biological products, more so than what the competition does. My focus wasn't to understand if we could replicate the lab level in another lab setting, but it was to take what they took at the lab setting and take it out to the field and to see, can we replicate this? Can we quantify this? Can we see this? And I think the thing that to me is so impactful is listening to our microbiologists talk about potassium and zinc, for example. These are two capabilities that we didn't add any more strains to the 401, but it was simply because we were taking soils and tissues and we were doing trials. What they saw was we had more potassium where we used Environoc 401. Continuously, whether that was against a different biological product or whether it was just simply against an untreated control check. And so what they did was they went back and they developed a media to be able to test, do our microbes have the ability to solubilize potassium and zinc? And what they found is that we did. So, you know, it was not only was from my side of things was trying to quantify and qualify, you know, what we saw at the lab level, can we replicate it in the field? And the answer is yes, we did. But we also found expanded capabilities because of the soils, because of the tissues. And I say that to say that we're not done yet, right? We're continually looking for other nutrients. Can we quantify what the nutrient solubilization side of thing is, whether that is in a lab level setting or whether that's out in the field? I'm not going to get into a lot of yield data through my part of the presentation. Grant's going to do that, but I think this goes with really the next two slides really importantly. So this data comes to us out of Canada. The next two slides really do. And what you can see here is just the yield data that comes when the grower uses Environoc 401. So if you look at it, you have an untreated check, so which would just be the grower standard, showing everything on the left, what was applied, the pounds of nutrients, etc. What you have there in the middle is the addition of 20 gallon the acre of UAN. And then on the right-hand side, where Environoc 401 was used, was simply looking at when we put Environoc 401 without the addition of the 20 gallon an acre, what do we see from a yield response? And what you can look at there, and I'll let Garth talk about this a little bit here, is the p-value, the importance of the p-value, and how that becomes significant in this.
Yeah, so p-value is just basically a statistical standard that shows the quality of the data and the rigorous aspect. And again, this was a third party doing this independently. And I just want to kind of emphasize what Bodie just said, you know, from the nutrient efficiency piece. You know, on the left there, you see it's a traditional nutrient load. You got your MAPs, you got your DAPs, you've got all of your nutritional components. And then you've got the addition of UAN as a side dress, which is common practice. And then you have just adding 401 in furrow with that seed without the addition of that UAN. And if you think about the value of this, it's the yield data for sure. You know, we saw an increase with the yield, and we see that when you introduce 401 into that system, you start that association with that seed, with that root zone, with that soil, that nutrient release. But for me, what's really valuable here is think about the bushels that were gained by not adding that UAN. The profitability just from not having to make that pass. This would have been profitable even if the 401 treated area would have done 192 bushels. It did 225. But if you look at it from a manufacturing perspective, as a grower, you're taking a seed and you're trying to produce more seed. You add nutrients to that system. With the right biology, with a focus on that soil, you can get the same amount of yield potential with less nutrition. Earlier on, Bodie was talking about phosphorus, the ability of our technology, our community of microbes, to make unavailable phosphorus that's sitting in that total pool in your soil available. So you don't have to waste that dollar and get 30 cents from it. You can get that value from the soil since you've already invested it from previous applications. It's just not available. And again, this shows the diversity of our approach, whether it's phosphorus, whether it's nitrogen, whether it's potassium. By optimizing that soil system, by introducing a technology that is a community of naturally occurring beneficial microorganisms, through that addition, you can start making subtractions on your nutrients. Why? Because you've got a much more efficient system. You're getting more value from the nutrients you're adding, but more importantly, through the creation destruction model of microorganisms, you're releasing nutrients that are unavailable in the soil. I mean, it's breathtaking when you think about it. If you focus on the soil, you focus on the health of the soil, you can drive profitability. You can drive nutrient efficiencies. You can drive yield potential. And if you look at it as a system, there's opportunities to pick up bushels even before yield by optimizing the use of the nutrients you are adding, and in most cases, reducing the nutrients you are adding because you have a well-functioning soil system, i.e. a stomach for that plant, creating that available food that is currently in that soil in unavailable form. So I think that that's really important when you look at it. You look at it as a system, and you're looking at efficiencies. And our suggestion would be by focusing on the rhizosphere and specifically the star players in that rhizosphere, which are microorganisms, you've got a lot of opportunity to drive profitability in your current systems.
Thanks, Garth. This is taking a look in a controlled setting. So this is a test that was done in the lab. And really what it was, so you can see up here, it's a controlled environment, third party. There were 6 flasks used, 3 controlled and 3 treatments. You can see where they were incubated. They can see how many samples were taken. And you can see how the processes, the ICP machine, for testing and documenting the calcium and phosphorus concentrations over time. Really what this is looking at is the amount of nutrients made available in a 24, 48, 120, 168 hour, and 336 hours, so 14 days. What are we seeing in a nutrient solubilization, right? So now we have it from you've gotten to see the nutrient solubilization that we have talked about from the capability chart that I showed with 401, right? The 6 strains of the phosphorus solubilization. You've gotten to see a little bit about how it performs out in the field. And now you're getting to see it from a third-party verification. And you think about it, it's not just this year's fertility, it's the year before, the year before, the year before, it's 30 years before. It's manure applications, right? And the amount of nutrients that are applied that are in our soil that we're going after. And so you look at the nutrient level impacts of this. And this is something that we've seen time and time again, right? So when we went through the capability chart, what you saw was phosphorus capability, right? The ability, it said phosphorus solubilization, the ability to produce the phosphatase enzyme to break down and create unavailable phosphorus into available phosphorus. Go back to the beginning part of our presentation where we talked about all of those counter ions that could form complexes with those phosphorus, for example. And I gave the example of calcium ditriphosphate. What I think is fascinating here is we saw a 484% increase in soluble phosphorus, but we saw a 601% increase in soluble calcium. Right? And again, what did I talk about? Plants don't take up complex bonds, right? They don't take up calcium phosphate. They take up phosphorus and calcium in a plant available solution. But because of using a microbe that is focused on phosphorus and that enzyme is produced, that enzyme, think of the enzyme as scissors. What did it cut? It cut the complex. It cut the calcium phosphate bond and gave you calcium and phosphorus. And the reason I share this is because this is something that we have seen from multiple soil tests, thousands, hundreds of thousands of tissue samples. When we used the microbes, what we saw was a compounding effect. It wasn't just phosphorus. It was phosphorus plus calcium, phosphorus plus magnesium, phosphorus plus manganese, phosphorus plus zinc, etc., that we saw benefits from that. And that's really what it is. Biology at its core is understanding, number one, the problem that we are trying to go after, right? And some of that is nutrient efficiency. And some of that might be nutrient efficiency of phosphorus. Some of it might be potassium. Some of it might be nitrogen, etc. But understanding that there are secondary benefits because of this, right? So again, what you can see here, and this is obviously recorded, we'll make this available to you as well. But what you can see is what was the percent increase at that particular time, right? So at 24 hours, we had an 11% increase in phosphorus availability versus the control. At 48 hours, we had a 139% increase. At 120 hours or 5 days, it was a 250% increase, right? 290 at 7 days. And by 14 days, a 484% increase in our plant available phosphorus. This was a study that was just done 4 weeks ago that we got the results back. And as Grant was putting this presentation together, I said, man, this has got to go in this presentation, because I think for me, it speaks volumes of what we have demonstrated in the field at the lab and now third-party verification of turning a unavailable, not plant available, right? Not a plant available nutrient into a plant available nutrient by simply influencing the biological activity with microbes with a specific set of those capabilities to make these nutrients available. This right here, and I know that we're kind of running tight on time, so I'm going to try to go through this as quick as I possibly can, but not miss anything from an information side of things. So you've heard a little bit about our 401. Here is 501. Basically, if I was going to summarize what 501 is, okay, 501, that's the creator that we talked about, right? That's the creator. What we have seen with our internal data is that when growers use a 401, 501 system on the same acre, now we can talk about at the end of the presentation, how are the multiple ways that I can deliver these microbes? We'll talk about that. But what we have found, the ultimate importance is when we're stacking a creator and a consumer together, our growers' profitability goes up exponentially and our nutrient solubilization goes up exponentially. And I think that it's extremely simple from an explanation standpoint. Number one, we're working on that organic matter with, again, the poor R value to biological activity to convert that into the available food source, right? WEOC. Water extractable organic carbon. So what are we doing? How are we doing it? You're taking the cellulose, lignin, and chitin. So you can see that up here. You see those three are right in the middle, right? We have 2 strains focused on lignin. We have 16 strains focused on cellulose degradation, and we have 10 focused on chitin degradation, right? Cellulose, lignin, chitin, 2 of the 3 of those are the most complex carbons that exist in our soil system from plant and animal waste, right? So that's all of our residue left over, whether that's wheat, whether that's corn, the fodder, everything that is left over has a tremendous amount of cellulose and lignin in it. Manures that we apply, right? Cellulose and lignin, tremendous amount of that. And what we're doing is we're breaking down that unavailable pool into making it a available pool that then allows the 401, which you've seen the nutrient solubilization side, allows them a longer period of time in which they can facilitate or create enzymes that facilitate the nutrient availability. And that's what this is. This isn't about, is it a way to sell more product? It absolutely isn't. It is what we have found. Before I got here, it was 501 was used as a residue degradation, and that was about it. 401 was used in furrow or as a seed treatment, right? Environoc ST or 401 in furrow, maybe in a 2x2, but that was really it. And what we saw through using the soil and tissue testing platform was if we put the 2 on the acre, and you'll see some slides of this that Grant will go through, you see the yield go up exponentially as a grower. From an agronomist perspective, what I saw was our nutrient solubilization capabilities went up exponentially. And that's what my focus is on, is how do we create enough of the food source to feed the consumers that create and facilitate that nutrient solubilization for a longer period of time. Because and this is what happens, right, is if we don't put those creators in that soil system to break down that complex carbon or the unavailable carbon, what happens is you'll see a spike in biological activity, but then you'll see it tail off because you don't have the food source there. You don't have enough of that WEOC or the available carbon to be able to provide season-long biological activity and enzymatic activity as well. Again, this is just some field data showing the treated versus untreated in phosphorus in the top, potassium there in the middle, and just the available dollars per acre, what that means as far as the nutrient solubilization. And again, this is around our 501 or our meltdown technology. This is data off of my own home farm. I tell this story quite a bit. About 7 years ago, we had been soil sample every 2 years. We were fertilizing basically whatever the co-op recommendation was. That equated to about 185 pounds of MAP, 310 pounds of potash. Take a soil test 2 years from now, and it was about the same exact recommendation. And I farmed with my dad and my grandpa at the time, and they were just saying, man, it doesn't feel like we're making headway, right? We were supposed to be variable rating, and the reason we were going to be variable rating is so that we could put more fertilizer on our low spots and pull back some of our fertilizer on our high spots, right? And in that time frame, I was coming back from college, and I started, just kind of tissue testing. And as random luck would have it, I took a tissue test from a high testing phosphorus soil testing area and a low soil test phosphorus area. And I actually had more phosphorus in the low soil testing area than what I did in the high, right? And so you think about this, the have we been told wrong? What this comes from is there's the mindset of if we don't put a removal rate on, a minimum of a removal rate or a build rate, we're going to mine our soils, right? We're going to go to zero. I've looked at 100,000 soil tests. There's a couple of things that I have to admit. I've never seen a negative number on phosphorus, and I've never seen a zero. I've seen some 2s, some 3s, some 4s, but I've never seen a 0 or negative. Well, if you think about that and you think about removal rate, well, for me, I should be at 0, right? I should be at 0 or potentially negative with the bushels that we've raised, the amount of phosphorus and potassium that we have removed versus what I've applied. So in 2019, we had a field average of 217. In 2020, it was 221. And in 2021, it was 211. Well, what you can see is respectively 76, 77 and a half, and 74 pounds elemental phosphorus, right? 227.4 pounds of elemental phosphorus in a 3-year period. And I've applied 0 pounds in that 3-year period. I have, on potassium, I've removed 162. I've applied 135. If you look at this next slide here, what you'll see is zone data from my soil test, right? So 2019, we'll just look at zone 1. So the very top line there, we had a Bray phosphorus reading of 16, removed 217 bushel corn, didn't apply any phosphorus. The next year, my soil test level actually went up 8 part per million. The following year, it went up another 4 part per million. So in a 2-year time period, it went up 12 part per million, right? You look at what happened to our potassium. My potassium was 119. The first year went up to a 140. The second year went up to a 141, right? We were actually seeing nutrients move up on our soil test while we hadn't applied any phosphorus to the soil, right? And really, what does that equate to? Well, we broke it down here in this next slide as far as what the dollars per acre meant, right? So on average, our Bray values went up 17 part per million. If university standards are correct, it takes 20 pounds of P2O5 to move it up 1 part per million, which equates to 653 pounds of MAP. 1152 at the time was valued at $800 per ton, which is a $261 an acre value in phosphorus that went up, that all I did was using biological activity. I used meltdown and I used 401, and it allowed my soil test levels to continue to go up, to the tune that if I tried to fertilize commercially to achieve that, it would have cost me $261 an acre, right? Our potassium went up on average 6.5 part per million. Standard universities are correct. It takes 7 pounds of K2O to move 1 part per million. That equates to 75 pounds of potash. Valued at $800 a ton, was a $30 an acre in value. If you add in the fact of crop removal, right, that's 436 pounds of MAP for a value of $174.40 an acre. Potassium crop removal was 162 pounds. That would be 270 pounds of potash, but we only applied 135 for a net gain of 135 pounds. Again, a value of $54 an acre. So in 3 years, the gain on a soil test was $519.40 an acre of net gain, right? That we didn't apply anything. The reason that I want to show you this, and the reason that we want to talk about this, again, we talked about, I'm not telling you that you're never going to put phosphorus on again. I'm not telling you never going to put potassium on again. What this is really designed to do is to equip you, number one, with some education and information that this has been done before you, and maybe on some extremes, right? Am I telling you cut out all your phosphorus? No, I'm not going to tell you to cut out all your phosphorus. Again, we talked about it. We're not anti-fertilizer. We're pro-farmer efficiency. What I was doing at this time was I had seen my grandpa and dad go back and forth about the money that we continued to spend without the actionable results that should have went along with it. So I tell people, I was coming back from college. There were 2 things in my favor. Number one, I was smarter than anybody else because I just got back from school. And number 2, I didn't have any risk because it wasn't my name on the check. So I was willing to do a 180. They trusted me. They went along with it. In that 7-year time frame, our average yields have went up 40 bushel the acre, right? They've went up 40 bushel the acre on our field averages, not just a field. So what I want you to understand is that we're actually on our farm going to start implementing some phosphorus back into our soil to make sure that I'm not limited for the next 40 bushel gain that I want to have, right? Now, does that mean that I'm going back and putting crop removal on? No, the answer is absolutely not. I'm still using biology. I'm still promoting biological activity. What I'm trying to do is to make sure that as I try to go from 240 to 280 bushel corn, we know that the nutrient demand is significantly higher, that I make sure that I am supplementing biology and phosphorus to make sure that as I go up the next 40 bushel, I don't leave any bushels on the table. The reason I share this is, again, this is real world data. This is straight off of our farm. It's $519 an acre that I would have had to spend in commercial fertilizer to try to achieve these values. And what I did was simply use 401 and 501 on the same acre for a 3-year time period.
All right, thanks, Bodie. So I'm going to go
All
through
your-
some of the case studies and trials using our products. I'm going to start with Biodyne seed treatment. So this is the results of a 10-year data set on Biodyne seed treatment. And as you can see, there's an average of 5 bushels increase in corn and a 3 bushel increase in soys. But remember, it's important to note that yield does not tell the full story. And I think Garth and Bodie did a fantastic job of highlighting this earlier. Seed treatment promotes a better stand, uniformity, and faster emergence for your crops. And in turn, these fundamental benefits ultimately lead to a higher yield. Now, this is also Biodyne seed treatment. This is a Beck's independent seed treatment trial and was performed in Illinois. And this was over the course of 2 years. Biodyne ST was put up against 37 other competitive seed treatment products. And as you can see, Biodyne ST was the top performer, and it had a 6 bushel advantage over the average. So we placed number 1 out of 38 products total, and we are also one of the cheaper options on the market. And I truly think that speaks volumes. Now this is moving on to 4.1 and 5.1 technologies. This is a data set taken from a trial back in 2021. It included 30 different farms across the state of Iowa. The treatments were 16 ounces rate of 4.1 in furrow and a 32 ounce rate of 5.01 post-harvest. As you can see on the chart, with the addition of these 2 products, the available phosphorus in the soil was significantly higher. The increase in phosphorus represents what happens when you introduce a healthy, more vigorous soil system from our products. This is a 4-year case study performed in central Iowa. The data set shows 401 and 501 as individual treatments compared to when they are combined on the same acre. And in year 1, you can see that 401 alone and the 401-501 combination did much better than the untreated and the 501 alone. Year 2, there was a severe drought. I believe that the rainfall in 2020 was 17 inches behind the 10-year annual average. So it was really severe drought. As you can see, that is reflected in the yield results here. But even with the severe drought conditions, Meltdown 401 did over 10 bushels better than the untreated. And, it has been shown year after year that our biology performs best under stress. And I think that this is another good representation of that. 2021 was another good year of results from the biological teams. But as you see in year 4, you can see that 401 actually performed worse than the untreated. Now, why is that? Well, after 4 years of only using 401 on that test strip, extracting carbon from the soil system, as they were saying, to feed the plant, the total available carbon is being depleted from the soil, which after several years can begin to have detrimental effects on and you can see that here. And as Bodie had highlighted earlier, the 501 product is what breaks down that organic matter and restores carbon back into the soil system. And I would say that's why it's important to have these two microbial teams utilized on the same acre to maximize your profitability year after year after year after year. This is the last data slide. This is a trial from 2019 from a grower in Iowa. He's been planting corn on corn for 14 years, no-till. He applied 32 ounces of 501 in the spring and 16 ounces of 401 with the 624-6 starter. He obtained 9 more bushels per acre with the 401-501 together over the 401 biostimulant alone. And you can see on the left here the plant growth difference at the V8 stage. It's become evident to me that a healthy soil system leads to a healthier plant, which then leads to better performance of that crop. And I think that's evident on these trials here. This is another slide highlighting our microbial team technology. This is the 401-501 capabilities together. And I cannot emphasize this enough. When these two teams are paired together in the same soil system, I mean, I don't want to say it's magic, but it's almost magical what really we can happen and how we can affect that crop. So that is actually the end of our presentation here. I'd like to leave it open to questions at this point.
All right, I will probably have to unmute everybody or ask everybody that they can unmute right now. If they have any questions, then they can chime in. So when you guys are temperature outside or in the ground to apply this stuff, what does that look like?
Yeah, so from a from a residue degradation piece, we'll talk about meltdown. What we try to do is we try to get 3 days in a row of 40-degree ambient temperatures. If we can get that, we've sprayed meltdown not based on a calendar, but based on the temperature. We've sprayed up until after Thanksgiving. We've sprayed actually, if we get the window, we've been 10 days, 2 weeks before Christmas that we've applied. So if you're looking for the residue degradation piece, meltdown in the fall, you want to reduce tillage, or you need help with that residue cycling from a fall application. That's what we're looking for. 3 days, 40 degrees in a row. If it's if you're trying to apply 401, if it's going out in furrow, obviously it's going to be made when your planter the planter pass is being made. 401 can be used in a broadcast application as well. And it can be mixed with certain herbicides. We have comprehensive lists that we could kind of help you with that. The biggest one to know is Gramoxone and our biology do not mix, period, at all. Please don't try it. Outside of that, from a pre-plant application side of things, Honestly, you're probably going to spray a spring burn down or a pre-herbicide pass, within a 10-day to 2-week period of when you're going to plant. So soil temps are going to continually to gradually rise. The important thing to know is that as temperature rises, biological activity is going to ramp up. So if you're looking for a visual response, the more warmth that you have, the more biological activity you're going to get from that application. Again, you do not want the product to freeze. 3 days in a row, 40-degree ambient temps is really what we would try to achieve on an application side of things as far as temperature, Andrew.
Okay, thank you.
One thing I did forget to mention earlier was that, the 3 products we highlighted today was the Biodyne seed treatment, the 401 biostimulant, and then the 501 stubble digester. But 501 is also considered meltdown, so the products meltdown and 501 are one and the same. I just wanted to clarify that.
Odie, I got a question. This is Doug up here in Minnesota. I've got high CEC, high organic, a lot of cattle manure was put on the ground. I've got a lot of calcium tying up my phosphorus. Will this help release that, get the calcium to release the phosphate?
Will be unbelievable what you'll see from it, Doug. This is where I think the word shine comes to mind because of the amount of, number one, the higher your CECs. Obviously, in a dry year, you guys benefit from that, right? Versus me, I farm a 7 to 12 CEC. But the bad thing, the thing that you guys struggle with is there's that many more cations and anions that have the ability to complex, right? And so there's more tie-ups that happen in those heavier soils. Add in the fact of cattle manure, which typically, from a saturation side of things, has a tremendous amount of phosphorus, has a tremendous amount of calcium in it. Yes, those tie-ups are happening on a regular basis. So a 401-501 combination on that acre will help solubilize carbon as well as solubilize calcium and phosphorus at extremely high clips. We would have a lot of data that would be similar to that. The data that Grant actually just showed. What we didn't share is that it was 14 years of corn on corn. All of the stalks were baled off of that. Then they were used as a bedding in a Quonset hut for hogs. And then they cleaned that out and they spread the bedding plus the hog manure back across the field. So it would be 14 years in a row of that same exact system of manure being applied every year, and the amount of nutrient availability that comes from that, and the amount of pounds of available nutrition behind the biology is just unbelievable.
Okay. Yeah, we did took the stocks off 10 years corn on corn, put the manure back on, and that tied it up. I was looking at, I fertilized last fall for NP and K, approximately 200 bushel an acre. Got a goal of 225, 250. I was going to use 1034 in the furrow. With the 401, would I be able to leave that 1034 out?
How many gallons of 1034 were you looking at running?
Either between 5 and 10 gallons an acre.
So what I would tell you is I'd cut it in half. The reason for that is in your climate, and I assume that you're somewhere in the northern part or central part of Minnesota, where you have a chance that your soils are not as warm early when you're going to the field, that the response to some of that banded phosphorus can still be extremely important from driving ATP and rooting and getting that crop, that stand establishment. But cut it in half and put 401 in, and you will absolutely see an economic efficiency and a nutrient efficiency that goes with it. So if you were at 10, I'd say anywhere between the 3 to 5 gallon mark. If you were going to run 5, I would say again, whether you add 2 gallons of 1034, 2 gallon of water and a pint of 401, you will like what you see.
Thank you.
Say, what's the rate on seed treatment?
So the rate is an ounce and a half to 2 ounces per 50 or 3 to 4 ounces per 100 unit or per 100 weight. I mean,
And is it compatible with existing seed treatments that are already on the seed?
Yes.
yes, yep.
Okay, thank you.
When it comes to the 501, if that when it comes to effectiveness of it versus fall application, or if you were to put it in with your pre-emergence pass, what's the difference on fall application or spring application with the 501 as
Yeah.
far as benefit?
Yeah, great question, and thank you for asking that. So, where we talk about fall applications, an awful lot are for growers that are either in a, they want to reduce a pass. So some folks have to run a VT, or they got to chop stalks before they can get their chisel through it. Or folks that want to no-till, they want to reduce a pass or reduce all passes, and they're looking for that residue degradation. That's when we would want to use it in a fall application, and you get the nutrient solubilization as well as the residue degradation. In the springtime, you're still absolutely getting the nutrient solubilization that we talked about. You're still getting that WEOC conversion that we talked about. But what you're not going to get, because there's just not enough amount of time before the planter, I should say, before the planter gets there, is a noticeable residue degradation piece, right? So if you apply 501 or meltdown in the spring, it's because you're looking for some carbon benefits, some nutrient solubilization benefits. If you use it in the fall, what you're looking for is both of those plus the residue degradation. So on my farm, meltdown gets applied on every stalk acre in the fall, and the bean stubble acre, it gets applied in the springtime.
With some of these new carbon credit programs, are you doing a carbon test on the soil then too?
So we are testing. We are testing carbon in our soils. Every soil test that we take, we are getting a report of carbon. Now, a lot of these the carbon credits, the CI score seems to be the new one tied in with 45Z. Those aren't really looking at a carbon number. What they're actually looking at is trying to assign how efficiently. So the new one, right? Carbon intensity score tied to 45Z is, can you reduce your synthetic dependency, right? Can you reduce some nitrogen? Can you reduce some phosphorus? Can you reduce some potassium? Can you reduce passes diesel fuel through the field? And that all has a factor associated with it that helps elevate what your CI score is. The carbon credits, most of the carbon credit companies that I have had meetings with, a lot of them are the same exact way. Use a cover crop or don't use a cover crop. If you use a cover crop, it's worth X. What we are doing is we're looking at WEOC, and in our opinion, a lot of these companies are failing to be able to do this. Or to do it with a metric of showing a improvement in the soils. What we know is the more carbon you have, so the more carbon that you can make available, the more carbon you're actually going to be able to store. So as we can drive those WEOC scores higher, your ability to actually store more carbon in that soil goes up exponentially. But nobody, to my knowledge yet, is taking a WEOC score and assigning a direct value to that to give you a quantifiable number as far as an economic return on it.
This is Jeff Holland from J&J Agronomy, Northwest Minnesota.
Go ahead.
Is there and I apologize, I was in and out of here, but is there a preferred way to apply it? Is it on seed better or in furrow better?
Yeah, that's a great question. So one of the things in our early days as an organization, really we had 401 for in furrow, And what we quickly found out was that not everybody has in-furrow and not everybody wants to have in-furrow, right? So as a company, what we did was we decided that we needed to make products that fit into the grower's box rather than making a grower fit into our box. So between a seed treatment and a 401, in my opinion, they're doing two different things. They're doing similar things, but they're also doing different things. If I'm going with a seed treatment, what I'm trying to do is protect that spermosphere around the seed, right? I'm trying to prevent some of that abiotic and biotic stress directly around the seed to encourage and facilitate that emergence and that uniformity as fast as we possibly can, right? And you think about seed treatment, then you think about, as a plant starts to germinate and it starts to put roots and shoots, well, then it transfers somewhere around that V3 time frame is where it will transfer into the secondary root system, which actually becomes the primary root system for the plant. So it's doing two things, in my opinion. So I can't say that if I run a seed treatment, I would not consider in-furrow. If I run an in-furrow, I wouldn't consider a seed treatment. And I don't know that they're mutually exclusive. So I know that's probably not a great answer for you. Both of them have provided some very extreme value and benefits as a seed treatment and as a in-furrow. But if I'm going with an in-furrow, I'm looking for more of the nutrient solubilization that we talked about. And the seed treatment, I'm looking at it from more of a germination and early season stand establishment. Does that help any?
Yeah, so you could take your, say your cost per acre. I don't know what that is, but so I don't know what the rate is either. But what if you were doing a seed treatment and then going in furrow also?
Yeah, so I think you could do that, an ounce and a half per 50 or 3 ounces per 100, and then come back with so the full rate of 401 in furrow would be 16 ounces. But I think you could come back with an 8 to a 12 ounce use rate on the in furrow side. So that the economics would all potentially shake out, in your favor as far as what your economic input would be. And get the best of both of those technologies for what they're doing, the stand establishment, then transferring over to the nutrient solubilization.
Okay, yep, that's my question. And then I got, and if I missed this, I apologize too, but there is no liquid fertilizer. It's compatible with all liquid fertilizer?
It is compatible with liquid fertilizer, yes. 1034.0, 723.5, 624.6, UAN, ATS. One of the things that we talk an awful lot about is we'll use a just-in-time delivery mechanism is what we talk about. So what we want to do is we want to introduce the microbes into whatever fertilizer or whatever carrier they're going with as close to the application point as possible. We've done viability tests over an extended period of time, but what we're trying to do is we're trying to make sure that we're delivering the highest CFU that we can as close to the time of application so that when they go into the soil, these are all live, they're not spore formers, so there's no animation period. So the moment you put them in the soil, they're going to start to build their numbers and provide those benefits from you. So the just-in-time delivery mechanism is to make sure that we're delivering maximum viability that we can to that soil, to that rhizosphere, as close to application point as possible.
Okay, that all makes sense. Thanks.
Okay, with that in mind, how soon do you got to get that seed planted once it's treated?
So from a seed treatment standpoint, you're going to have, I will say 90 days extremely comfortable. We have done tests it was a product called Obvious Plus that was used as a fungicide insecticide. And we did the whole point of that was to answer the questions, are the microbes still alive 240 days after? And that was actually a year-long test that we still had viability after a year of our microbes not being mixed with a food source, not being put into the soil, where they wouldn't have a chance to grow their numbers, basically seeing, do we degrade the numbers? And we did that all the way out to a year. So 90 days after seed treatment, for absolute certainty, I have a lot of comfort with. Depending on what seed treatments you would start to stack in on that, would probably dictate our answers as far as looking longer, 120, 150, 180, 240 days, etc.
Okay, thanks.
So did you say 401 in furrow is compatible with insecticides or is not?
It is.
Is
I don't know if Zoom is going to kick us off at the hard stop at 2:30. So if it does, thanks everybody for joining us. But if there's any other questions, we'll just go as long as people have questions.
Yeah, I got another question. As far as liquid manure application, can you add this product to a liquid manure application?
Yes. Yeah, we have actually done that. So you heard a presentation around 401, 501. We have a team that would be called 301 that is designed for going into lagoons, going into hog barns to break down the solids to have a more homogeneous blend, make it easier for pumping and to be able to get more out of the pit. We have stacked 301, 401, and 501 together. We've mixed them ahead of time so that when they go and apply it, they're getting the benefits of the carbon solubilization. They're getting the benefits of the the plant growth promotion team. So the nutrient solubilization we talked about with 401, and then they're getting the the solid reduction, if you will, of the 301. So yes, we have absolutely applied these with liquid manure applications on a lot of acres.
Okay, thank you.
Yeah, and for the sake of time with all of this, we didn't have the 301 as part of today's presentation. If anyone has any questions about the 301 and the manure management portion of that, we contemplated doing something like this again that will be more of a focus on the 301, but reach out, call the office, and we'll reach out to whoever we need to and have those conversations with them.
Okay, I've got a question on the 501. When you spray that on the fall, can you graze cattle behind it?
Absolutely. We've
Yes.
done it for 4 years now. Yes.
Yeah, so just want to add this, 401 and 501 are OMRI badged, so they're USDA certified organic. And our seed treatment is organic as well. But yes, as Bodie said, we've had growers that put it down and then bring their cattle right back on, and there's no negative effect. There's a suggestion it actually helps with the digestion, but we're not going to claim that. We just
I was wondering.
we can just say that there's no effect on the animals.
Okay.
That's 3 days at 40 degrees.
All right. Anyone else have any questions?
You said you can spray 401 over top on the ground in the spring instead of in furrow?
Yeah, you can add it to a pre-emerge or a post-emerge. The rate will be a bit different, closer to a 24 or a 32 ounce rate. But as Bodie was saying, we've created our technology so that it's easily implemented into a current system and doesn't require any systematic changes from an operations. Obviously, the nutrient efficiencies piece can provide some changes, but no, and there's a lot of opportunities to, if you're doing a pre or a post, to do 401 and 501 together. Take advantage of that liquid pass you're going to make, and you'll kind of get the power of both of those teams in that pass.
So you're saying you could do a 401 in the fall?
Well, I'd say in the fall you want to do the 501 because you're looking for the residue breakdown. But as we're talking about an in-season pass, since you can't go in furrow or on the seed, putting that 401 and the 501 together is a pretty powerful combination in season.
How many gallons of water do you recommend when you do that? If you got the 401 and the 501?
Looking at a 10 to 15 gallon per acre. You just want to make sure you ensure good coverage. So depending on your rig, it may be a little bit less, a little bit more. But fundamentally, you're just trying to make sure you get a good spray across that ground to get good infiltration.
And probably like most, a nice rain right after would be a benefit, right?
Yeah, absolutely. Obviously, it's not a requirement, but you know,
Okay.
if you can time it before moisture, you know, that just basically creates that soil solution a little bit faster and allows all that good stuff to get down into that rhizosphere.
Right.
All right. Anyone else have any questions? All right. Well, thank you everybody for joining. Thank you, Garth. Thank you, Bodie and Grant, for jumping on and doing this for us and educating everybody. We really, really appreciate it. If anyone has any questions, please reach out to us. Call the office. Call whoever at Bass, and we'll get you guys you know, any and all the information that you guys need.